Hello and good evening. It is such a great pleasure to welcome you here tonight for our inaugural Longwood seminar for 2015. I'm Gina Vild. I'm the Associate Dean for Communications and External Relations at Harvard Medical School. Over the past 15 years, these seminars have enabled thousands of individuals to learn about the latest research and medical information that emanates from Harvard Medical School. They have been successful because of your interest. It's your attendance and more recently, your participation on our many social media channels that have made these seminars a success. So thank you. And I see so many familiar faces here tonight. Just a quick show of hands if you've attended a Longwood seminar in the past. Great, thank you for returning. And for those of you who are here for the first time, I'm hoping you enjoy the experience and that we see you back in the years and the months to come. I also want to say hello to all who are watching through live streaming. If last year can serve as a sampling, tonight we have viewers joining us from all over the world, including Saudi Arabia, Mexico, Canada, Colombia, Italy, and the UK. So welcome all. We select the topics, as you may know, for the Longwood seminars through crowdsourcing, which means you tell us what you want to know. So this past year, hundreds and hundreds of voters have weighed in through our Facebook friends and through email. So the topics tonight were what you told us was most important to you. So tonight, of course, high-tech medicine. On March 31st, understanding food allergies. On April 14th, music is medicine. And our final seminar on April 28th, diseases gone global. I know you're anxious to get to the program, so just a couple of announcements. And we should have a slide soon that summarizes some of what I'm going to tell you. So you will see information shortly. You will see information shortly on how to obtain certificates of completion and professional development points for teachers. We also want to invite the Longwood Seminar, you to view the Longwood Seminar website to see our past Longwood Seminar videos, and all of this year's videos will be put on the website soon after the events are held. And it's also a fabulous place for you to access additional supplemental reading material. We hope you will participate in our Twitter conversation by using the hashtags that you'll find on the screen. And thank you for turning off your electronic devices. Today's seminar is called High-Tech Med, the newest wave of medical innovation. Since its founding, Harvard Medical School physician scientists have been revolutionizing the way medicine is practiced and the way in which healthcare is delivered. From Benjamin Waterhouse's introduction of the smallpox vaccine in 1799 to the first public demonstration of anesthesia and surgery in 1846, the introduction of the electrocardiograph in 1914, and the use of the iron lung that helped polio-paralyzed patients to breathe in the 1920s. And these are just a few of the examples. Harvard researchers have employed novel methods and technologies to improve the health of people throughout the world, and they are curing disease. Today, our researchers continue this proud tradition, whether using their knowledge to make surgical and diagnostic procedures less invasive, or by developing more effective instruments and biomaterials. Our scientists are not only on the cutting edge, they are the leaders, bringing new technologies to the bedside of men, women, and children. And it is truly a privilege to be able to introduce you to a few of them tonight. We think of them as explorers, inventors, innovators, and caretakers. People who are shaping the medical terminologies of today, the technologies for tomorrow. So I'm delighted to introduce our experts. Dr. Connor Evans is a Harvard Medical School Assistant Professor of Dermatology at Massachusetts General Hospital. Dr. Jeffrey Karp is a Harvard Medical School Associate Professor of Medicine at Brigham and Women's Hospital. But first you'll hear from Dr. Elazar Edelman. Dr. Edelman is a Professor of Medicine at Harvard Medical School, a senior attending physician in the coronary unit at Brigham and Women's Hospital, and the Thomas D. and Virginia W. Cabot Professor of Health Sciences and Technology at MIT. Dr. Edelman directs the Harvard-MIT Biomedical Engineering Center, dedicated to applying the rigors of the physical sciences to illuminate the fundamental biological processes and the mechanisms of disease. Please, let's give all of them a warm welcome, and thank you.
Thank you, Dean Bills, and welcome to all of you. It really is my honor to begin this and to talk with you. So this isn't going to be a lecture. We're going to have a discussion about why and how innovation makes medicine today a medical science and makes it a fertile field. So these are my disclosures. They're not financial disclosures so much as disclosures of people that I work with in industry to help us realize the potential of all that we work on. We work really, and we live in an amazing time. In our lifetimes, irrespective of what state you live in, irrespective of whether you live in a state where virtually no one smokes to one where everyone smokes, to whether your meal is fish or a cheeseburger, the incidence of cardiovascular death has dropped sixfold over the last 40 years. Not 6%, 600%, sixfold. And even for lesser diseases, I'm a cardiologist, so lesser diseases like cancer, you still have found that in men and women, the incidence of cancer death has dropped by a fifth to a seventh. We live in a marvelous time. And the question really, or the questions are twofold. One is, how did this come about? And two, how do we make sure that it continues to happen? Anyone know who this man is? Dick Cheney. He's a hero to us in medicine because Vice President Cheney has had 5 myocardial infarctions. He's had bypass grafting. He's had endovascular stents. He's had angioplasty. He's had an implantable defibrillator. He's had saphenous vein grafts. He's had pacemakers. And he had a heart transplant. And so, irrespective of whether you agree with him politically or not, he's a poster boy for innovation in cardiology. This is an electrocardiogram, not his. It's any one of my patients, a patient with an acute myocardial infarction. And when President Eisenhower had his myocardial infarction, he was advised to go to bed for 6 weeks and snuggle with Mamie. I don't know who got the worst of that. But in any case, his chance of dying from his heart attack was 50% before he made it to the hospital and 50% thereafter. And today, though it's still the case that half the people who have a heart attack will die before they reach the hospital, only 5% will die. 95% will survive. In fact, here's the incidence of cardiovascular death. It's dropped dramatically. So who discovered the coronary arteries, the arteries that surround the heart? Anyone know? So Leonardo da Vinci. It was Leonardo da Vinci almost 500 years ago discovered the coronary arteries. But it took 500 years before, because really modern cardiology starts at the time of Eisenhower and it peaks at the time of Cheney. And the question is, what drove us, number one, and what took us so long? This man defined the circulation in 1600. This man coined the term angina pectoris in 1770. This man, the man who incidentally discovered sickle cell anemia, was also the first person to diagnose a heart attack before it occurred. So why? Why did it take 500 years to go from here to here? Anybody? What did we need that we didn't have? Yeah, we didn't have technology. We didn't have electrocardiography. We didn't have x-rays. And that's why innovation is still important. More in my lifetime, how many people here know someone who's had bypass surgery? How many people here know someone who's had a stent? How many people here know that all the commercially available stents were developed and evaluated in the Harvard-MIT system? So when Vice President Cheney had his bypass surgery, he had a 91% chance of not having any problem up to a year. Around that time that was happening, people, especially in Emory University, were inventing balloon angioplasty, the ability to stick a balloon in an artery and blow it up and open it up. And it worked great, except that 40% of patients within a year would have to come back for something else. In and around that time, Campbell Rogers and I, working across the street, Building C and Building D, started to ask basic questions about what the processes were that drove this. Now, at that time, this was the first section of an artery that was ever double stained. It was stained with two things. One that showed that it had a lot of smooth muscle cells, and the other in brown, a lot of proliferation. And the thought was that this was a proliferating lesion, and I'm not going to go through this in great detail, but what we showed was that whatever was going on was incredibly complex in time and space and cells and molecular targets. And it took us years to define exactly what was going on, except we eventually figured out that there was clotting, and there was inflammation, and there was proliferation, and all of these things were critically important to understanding what happened when you put a balloon in an artery or you bypassed it. What we suggested and what we worked on was that it was better not to just blow up a balloon, not just to take something and stretch the vessel, but to put something in and leave it there. And we worked on a variety of designs of stents, and yes, with our colleagues in industry, because you can't innovate without making something that has a lasting impact. And we eventually help people design and develop these devices, which are stents, metal mesh tubes that stay in an artery, and unlike a balloon, which blows things up and then recoils back down, permanently deforms the vessel. So when Vice President Cheney had had the benefit of his angioplasty and had to come back with a problem, he got a stent, and the 40% failure rate rose to 75%. The nice thing about being a professor, and Professor Evans and Karp and Dean Violet will tell you this, is that you work with brilliant people. And the most important thing is to not say anything when they walk in your room with a great idea. So I always, when I talk with my students, keep my hand in front of my mouth because I'm afraid I'll say something stupid. So when David Wu and Chao Wei Wang came in and they said, you know, if we put drugs on a stent, we could deliver them right to where things go. And so this was one of the first drug-eluting stents. We made it, if you will, in our garage. We dipped a stent into a solution with orange drug. And it looks kind of woolly, and it looks kind of horrible, and you wouldn't put that in anybody. But we actually showed that when you put it in, you actually imprint deep into the vessel drug. This is 200 microns deep into a vessel. You stamp drug into a vessel. And that opened up an era of incredible innovation where whole schools of people were now making these devices so that when this got made in an industrial setting, our woolly mammoth stent looks like this. This is a metal stent with a coating 2 to 20 microns in thickness that can release drug for up to 3 months. And when you put it in, now, as opposed to a failure rate of 40% or 25%, the drug-eluting stent does even better than the gold standard bypass surgery. Now, the truth is that innovation usually starts in academic settings. Name the top 100 innovations in the last century, and I will tell you that virtually all of them arrived or were devised at some university. the electrocardiogram at the University of Leiden and X-rays at the University of Würzburg. Insulin was synthesized, if you will, at the University of Toronto, and pacemakers and angioplasty in the human genome. Everything that you will hear from the speakers to come about detection, the diagnosis, the design of drugs and devices was started by some student in someone's laboratory because people like you made it possible for them to do their work, and you explained why it was important for that to happen. It's no accident that the incidence of cardiovascular disease, the death from cardiovascular disease, and the death from cancer has virtually plummeted because we are in this amazing phase of innovation. We are in an incredible time where academicians are provided with the resources to make their innovations impactful. So I have a fundamental question, because this is all we think about. Who should get the Nobel Prize? And when I pose this question in front of my colleagues, I guess, oh, who should get the Nobel Prize? And then I put a red line through that, and I say, that's not important. And then they all sort of sit deflated, and they say, yeah, oh, yeah, right, it's not important. So the real question is, how do we make sure we can recapitulate this experience? Because there is, at the same time as being in a wonderful setting, a real crisis in this country. How do I know that? Well, I edit a journal. I edit one of the, there's this wonderful journal, Science Translational Medicine. I'm one of the advisors of the journal. And one of the editors, Kelly Lamarco, and then a colleague, Marty Leon, and I, we looked serially at all the papers on where innovation is going. And until 2000, there wasn't much of a concern, but in the last 15 years, there are now 10,000 papers on whether we are in this country suffering from a crisis in innovation. So certainly we're not suffering from a crisis in writing about it, whether there's a crisis, but we are suffering from an issue. So very quickly, because we really want to hear from our speakers, what are the issues? Well, this country spends virtually nothing on innovation. The GDP of this country is $14 to $16 trillion, and the life sciences budget is only $30 billion. Good companies spend about 10% of their net value on research and development. As a country, we spend 0.02%. This country has an amazing literacy rate where virtually all can read and all can write. But the truth is the number of people getting doctorates has plummeted. And in this country, if you start getting a university degree, then the likelihood that you'll actually get it is only 66%. Contrast that to what happens in Japan, where it's 92%. Look at the number of PhDs in engineering and science in China, and look at what's happened over the last 15 years in the United States. There are multiple barriers, and you're going to hear about these things. What are the barriers to innovation? Well, first, there's barriers to envisioning, conceptual science, innovation to renew, to alter, to make new, the introduction of new elements or forms, and the ability to realize those improvements for people's lives. Envisioning, implementing, conceptual science, engineering. I'm not talking about entrepreneurship. Look at the way I'm dressed. I don't care about money. If I did, I wouldn't be here. So I'm getting rid of that. That's not what we're talking about, and that's not what you're going to hear about. I'm going to skip over this for the sake of time, other than to say that there's a tension. We're not introducing as many drugs as we did before. We're not introducing as many devices as we did before. The time it takes for a new device to be approved in Europe and the time it takes for it to be approved in the United States has widened and widened. When the stent was first introduced, it was introduced 1 month earlier in Europe than it was in the United States. With the drug-eluting stent, 30 months. The United States was 43rd in the world in introducing the new heart valves that we work on between Argentina and Chile. Prima non nocere. Anybody know what that means? Used to be part of the Hippocratic Oath. Yes. means above all, do no harm. You can't do that in medicine sometimes, especially if you're an oncologist. Primensiere means rush to treat. So there's a tension. If you, above all, do no harm, then you never allow technology to come into the community. If you rush to treat, then you potentially leak technology too early. What do we do here? What do academicians do? Well, because it's Latin, it sounds a lot better if I made it, so I made my own Latin term. Primensiere, above all, seek to understand. And that's what you're going to hear about from Professors Evans and Karp. Above all, seek to understand. I'm going to leave you with some questions, and you're going to hear the answers to it. And then I'm going to make a pithy statement at the end, and you're going to applaud. So, are we funding the right projects? Are we working on the right things? Are we training people properly to be innovative? Is our educational system optimized for innovation? And how can innovation, science, and medicine mix optimally? You're going to hear about that from these wonderful people. Here's my pithy statement. Medicine without science is just an art form. And without innovation, it's simply sterile. I want to show you today by introducing first Professor Evans and Professor Karp how to make science and medicine, engineering and innovation mix so we have something that is simultaneously scientific and simultaneously fertile. Thank you. So now it's my honor and pleasure to introduce Professor Connor Evans, as you heard from Dean Wild. He is an assistant professor at the Wellman Center, Photomedicine at Harvard Medical School at the Massachusetts General Hospital. And his work has revolutionized photomedicine, our ability to image and detect, our ability to define events and to treat them as well using light we can see and light that we can't. And so with that, I'd like to ask Professor Evans to come up and take the podium.
Well, thank you so much for the introduction, and I, as a young faculty member at Harvard, I hope to live up to the hype of my introduction. I'd like to talk to you all about two things tonight that are very near and dear to my heart, and that is the topic of photomedicine, that is the interaction of light in tissue, light in cells, and how we can use photomedicine to solve problems. And instead of introducing the whole field of photomedicine, because I could do this for hours, trust me. I bore my students at Harvard constantly with this. I'm going to try to talk about three stories and introduce how we bring innovation to medicine with photomedicine. So first, let me jump into photomedicine itself. It's the title of my center, the Wellman Center for Photomedicine, and I'm sure every one of you have interacted with photomedicine in some way today, but actually have not even known about it. So what is it? It is when light interacts with tissues. So we take a ray of light, we take a laser, we zap tissue with it, and what happens? Well, a lot of things can happen, and that's the neat part about photomedicine. But in general, we can think about four major things happening. One that can happen is light can scatter. And scattering is something that you are all very familiar with. If you hold your hand up and you look at your hand, what you're seeing is light scattering off of your hand. It's the white creaminess, it's not transparent. And how light scatters, the way it scatters, and the information it carries, we can actually use to make diagnostics and to do very neat science with. Light can also emit, so a molecule can absorb light and then it can re-emit that light, and we can utilize that to tell something about our samples. And in fact, scattering and emission are the foundations of imaging, microscopy, and a lot of the diagnostic methodologies that we use. Light can also do chemistry, and in fact, the reason that you are seeing at all right now is because there's a chemical reaction going on in your eyes at all time. Light is striking your retina. It is converting cis to trans-retinol, and in doing so, you are seeing it conducts vision. So photomedicine occurs, and photomedicine can do some very neat things. We can have light-activated therapies, we can kill cancer with light, and we can also engineer tissue using some of our light. Now, what's very interesting is that we can also use light to do work. We can heat things. We can create pressure. We can create plasma. This is where we have laser surgery, where we have smart devices that surgeons can introduce that can make their lives easier, that can have higher resolution imaging, that can do a number of neat tools. And lastly, light can also have a negative consequence. And a large part of photomedicine actually is in studying the negative parts of this. If you walk out into the sun, I know we haven't seen much of it so far in the last couple of months, but UV light can give you a sunburn. It can generate cancer, it can cause mutations, and that is a large part about studying photobiology, photomedicine. So as my partner in crime, Jeff, will talk a little bit more about, a big part about what we have to do in medicine, and especially in photomedicine, is translating our tools to the clinic. We have to start somewhere, usually by my bench, and eventually we have to get to the bedside. And as was mentioned, that means we have to interact with a whole lot of people along the way. So rather than describe the translational pipeline, I want to illustrate it by sharing with you 3 stories, technologies that have come from my center, from the Wellman Center for Photomedicine, that are along this pipeline. So I'll talk to you a little about smart bandages, tools that we're building in my laboratory, about virtual biopsies, which are coming from Gary Tierney and his lab, and colleagues that are somewhere in between. And I'll start off with laser hair removal, which is actually photomedicine that you may not actually have thought about. So let's talk about laser hair removal. Now, contrary to most people's belief, laser hair removal was not started because people wanted to get rid of the little mustache or trim their bikini lines. It actually started with a request from the United States Army. So why was this? Well, there's a problem, and anyone in the audience who has a beard like mine, or people who have to shave parts of their body, know that we can get this thing called pseudofolliculitis barbae. What is that? That's barber's itch. That is these little pimples that can appear on your face. And anyone who's had a very close shave knows this can be a problem. Now, why is this an issue? Well, the U.S. Army requires their servicemen to shave close, have a very nice close shave. And especially for servicemen of color, this can be a problem because they have very curly hair, very curly beard hair. It can curl right inside, and it can cause ingrown hairs, and these can become infected. And this actually prevented soldiers from putting on gas masks and became an actual challenge during the original Iraq War because people couldn't wear their gas masks in the field, and they were ill-fitting. So the Army came by and said, can you do something about this? Well, the interesting thing is, yes, we can. Hair growth is actually pretty interesting. Hair growth passes through 3 phases: anagen, catagen, and telogen. Anagen is the phase that we all know. It's the hair that grows. Now, your hair normally does fall out. Hairs can stop growing and then are replaced by new hairs that grow in. So, you know, the hairs that you find on the floor and whatnot, that's normal. You're supposed to lose hair. It's part of the cycle. And a lot of work actually went into trying to slow down or stop the hair growth process entirely. But that was really hard. Now, when you're working with lasers, there's this tendency to try to think that everything can be solved by blowing things up. And it's pretty true in a lot of cases that you can solve problems. So one of the things that came to mind was, well, if we can't stop hair growth, can we eliminate the hair follicle altogether? Can we selectively go in and destroy just the follicle with very, very high precision? And the answer is yes, and absolutely that can be done. And the way it's done is by using one of 3 interactions. And you'll see this slide a few times. We can introduce light to tissue, and it can do a couple things. It can scatter, it can emit, and we're going to talk about absorption. Now, absorption is what gives things color. It's the reason that my shirt is blue. It's the reason the lady in the front has an orange sweater. Molecules absorb, and the light that does not absorb is what we see as its color. Now, we are all naturally, even without all our clothes on, carrying around chromophores, carrying around that molecules can absorb. And the main molecule that we carry around with us, unless you are a redhead, and I don't see many in the audience, is a molecule called, it's a melanin molecule called eumelanin. It's what gives our hair its brown color, what gives the dark hair colors, it's what gives us our skin tone. And the amount of eumelanin that we have is actually controlling the color that we have. Now, what's interesting about eumelanin is it's been designed over millions of years by evolution to be excellent at absorbing light, to protect your skin. And it's very interesting how it works. So eumelanin looks something like this. It's a polymer. It has lots of individual pieces that link together. And eumelanin is this enormous polymer that absorbs light across a huge range of wavelengths, all the way from the ultraviolet all the way out to the IR, beyond where your eyes can see. Now, what's interesting is, and anyone who has very dark hair who walks out in the sun can tell you this, is that when eumelanin absorbs light, it actually can actually emits that energy. It stores that energy and then emits it as heat. So yes, if you have dark hair and you're walking out in the sunlight, your hair is heating up actually from the visible light that's being absorbed. Now, what came to mind to Rox Anderson, to John Parrish and colleagues at the Wellman Center for Photomedicine is, could we actually do something with this heat? And the idea is that we have all these follicles. These hairs come down to this follicle. And in the follicle, on the edge of the follicle, are what we call stem cells. Now, stem cells you may have heard about. These are cells that make other cells. These are cells that renew the follicle. Different cells in the follicle will die all the time, but these stem cells will differentiate, it will grow, and regrow follicles all the time. These are the ones you have to get rid of if you want to get rid of the follicle. So how can we do that? Why not put a whole lot of heat in a small period of time in one location specifically? Specifically right here. This is all the eumelanin in the hair shaft. And so what was designed was a laser that could actually deliver light and create so much heat that it actually introduced a cavatonic shock wave. Just like one blows up a hot air balloon. You introduce heat and it expands. In the same way you can expand tissue. And laser hair removal works this way. You expand the tissue so quickly with a pulse of light that it actually kills all the cells surrounding the hair, including the stem cells. And then naturally over time, the follicles go away. Now remember I mentioned that hair grows in cycles. Only when there's a hair, only when hair is growing can you actually take out those follicles. So that's why you have to go back, if you have hair removal, to catch hair that is not in its active growth cycle. So it takes a few cycles. But that's how it works, and it worked very well. So this is actually the original laser. This is what it looks like in our laboratories. This is a ruby laser. There's actually a piece of polished ruby in here that creates the pulse laser. Here is a woman who came in for the initial study, had hair on her lip, and it has been removed. And thanks to translation, thanks to the involvement of doctors and physicians and numerous engineers, you can now go to your dermatologist and have treatment with these medical lasers. So this is something that is already in the clinic. It was an idea that started back in '94 and made its way all the way into the clinic over a period of years. And it's a cosmetic dermatology, so it's actually went pretty quickly. And the US Army has been very happy with their investment. Now, one of the interesting things about this project is it's very easy to see whether or not we've had success, right? If you look at a patient, is the hair there or is it not there? Now, in a lot of situations in medicine, what we want to look for is not as apparent. You can't see it necessarily. It's happening on the level of cells. It's happening on the level of groups of cells. And what we do normally in order to understand what's happening is we do a biopsy. And a lot of people here have either had a biopsy or known someone who has had a biopsy. And the problem with biopsies is we go in and remove tissue. Now, we can't remove tissue everywhere, right? You can't go, for example, it's very difficult for us to go in and biopsy the retina, the back of your eye. You wouldn't want to do that. You need that tissue. Biopsying the brain you can't do very often. Or maybe you have an area that's just so large, say your entire esophagus, that you can't biopsy the whole thing. Well, what people have been trying to do, and what we've been trying to do at the Wellman Center, is to create virtual biopsies. That is, ways of actually replacing the biopsy needle with imaging devices so that you can rapidly capture data at the cellular level, but do so very efficiently. And people can ask, why don't we use things like MRI, PET, CT, CAT scan, and ultrasound? And the very short answer is the fact that these tools have too low resolution. They can't see individual cells. They're millimeter scale. We need to get to micrometer scale resolution. And how can we do that? Well, absorption we've already used for something. Let's think about how one might use scattering. Now, I mentioned ultrasound. How does ultrasound work? We launch sound into tissue, and it bounces off of structures and comes back. And because sound moves pretty slowly, we can time how long it takes to go in and to come back. And that allows us to build up images. But light moves really quickly. It moves 3 times 10 to the 8th meters per second quickly, way too fast to time. But what we can do is we can actually race light against itself. And the way we do this is with this very neat toolkit called optical coherence tomography. And this was invented right down the road at MIT and perfected at MIT and at Mass General Hospital. And the way we do this is we race light against itself. You take light and you split it into 2 paths. You send one light down a race course where it just goes to one end and comes back. The other side is the race course that goes down into the tissue where it can hit different structures and bounce back to us. And then what we do is we see which one wins. And we do this in a cool little device called an interferometer. So we send light down to the sample, and it bounces off of different structures. and we send light down a reference. Now, the really neat thing about light is when you race it against itself and they tie, what ends up happening is we create interference. We create these little blips, these little fringes, and we can measure them with extremely high accuracy. And what we can do with that is that these blips occur only where the racetracks meet, only where you have reflections in the tissue. And in fact, we can take these and we can scan over tissue, and we can actually recreate the entirety of the tissue. This is actually my friend Heil Park's fingertip. This is the epidermis, the surface. This is the dermis. And this little squiggly line right there is actually the sweat duct in his fingertip. And we're seeing this on a cellular level resolution because light is interacting with those levels and bouncing back, and we're timing that. So how can we use this for virtual biopsy? How can we use this to look at large area of tissue? Well, one big problem that my colleague Gary Turney has been working out is the problem of the esophagus, specifically Barrett's esophagus. Barrett's esophagus is a pre-cancer condition that a lot of you may have or may have in the future. And it's difficult to diagnose because you have to look at the entirety of the esophagus, which is quite long, quite complex. And if you really want to know what's happening, you need to take a biopsy. But Gary is a pathologist, and he'd like to do better. He'd like to do his pathology in living people and not taking a look at the biopsy they're taken out. So he took this tool and with his colleagues actually created this. This is a little balloon. You want to talk high-tech med. This is a little balloon. And that little OCT scan, that optical coherence tomography scan, is happening as it goes round and around and around in a circle. And so this was designed, this goes down in the esophagus, and what they do while the patient's asleep is you simply tug it. So how does this work? You're going in a big circle, and you pull it. And so it scans a spiral. And it just spirals all the way up your esophagus. And so it collects, and what it can collect is something that looks like this. This is actually an image of a patient's esophagus as it scans all the way down. You're looking at every single layer of the mucosal layer. You're looking at the muscles in the esophagus all the way deep. And you can get all this information in real time as you do the pull. Now that's pretty neat. And this can reveal very quickly where you have regions of abnormal tissue, such as Barrett's esophagus. But I don't know about you. When I look at this, I really don't feel like having that down my throat, right? I have to be intubated. I have to be back. I have to be asleep when this is done. So Gary went ahead and did something even cooler. And that's to actually put all of OCT in a pill. I don't know if you can see it well, but he actually put all that tech in a little pill that's the size of a penny. And you can swallow this, and you do not have to be awake when it's done. So let me show you how this is the very first in man test. This guy is quite brave. What we're going to show right now, what you're going to see right now is actually the very first time this is done. Here's that's the little camera. That's the OCT pill. He's going to go for it. Yeah, that's what I would do. And he's going to swallow with some water. Now, it's on a tether. And at the end, you can pull it right back up out of the stomach. It's not a problem. And here it does. Now it's going to start spinning around in a circle. And as he swallows, as the natural swallowing motion occurs, it collects this data in real time as it goes down. And it can map the entirety of the esophagus. This is technology that's just about in the clinic. This is the kind of thing you're going to see in the next couple of years, and the kind of technology that's going to make your life much easier. Because at the end of the day, instead of taking biopsies, your clinician can take a drive right down your esophagus. So, really neat. This is all from my colleagues, Gary Turney, Brett Bouma, and others at the Wellman Center, who are building these very cool cameras, these very cool tools that I think are going to change medicine in a big way. All right, now we got started late, so I'm going to pretend that's green and move forward. So what my team is doing is a little bit different, and I like to spend a little time talking about this. This is structural imaging. This gives you the ability of actually seeing where things are. But if you want to take a look at the health of tissues, if you want to take a look at how things are recovering, say from a wound or from surgery, there's one really big parameter that's important to measure. And that problem is how to image oxygen. Now, oxygen, you know, is really important, right? If you hold your breath, if you stop breathing, if a patient doesn't receive adequate oxygenation, cells die, tissues die, and this is really critical. Oxygen supply is very important. But oxygen is odorless, it is colorless, and it is really, really difficult to detect. Now, granted, you can start sticking needles in people and doing things, but that doesn't give you the same capability. That doesn't allow you to see oxygen in tissues and the health of tissues over large areas. So what do we turn to? We turn to a mission, specifically something called phosphorescence, and I'll move through this a little quickly. Now, phosphorescence is something that you all know very well. If you ever had glow-in-the-dark pajamas as a kid, I know I did, the feet glue at night. If you've had watches, like your watch face can glow, these are all phosphors. And phosphorescence has a really neat property. So phosphors can look like this. This is the phosphor that I use in my lab. This is the phosphor right in the middle, and these are molecules that make it bind to things. So what's neat about a phosphor is that you can give it light, and then it takes that light in and it stores the energy. And phosphorus can be stable for quite a long period of time. It's why your watch face can glow for an hour or more. Now, the interesting thing is eventually it'll release that energy, and it can release it by emitting light, and that is the phosphorescence that your eyes see. Now, another way it can lose its energy is to oxygen. So oxygen can come by, and just like two people bumping into each other in a crowded hallway, oxygen can collide with the phosphor, and it can take away that energy. And when it does so, the phosphor can no longer glow. It can no longer emit anymore. So this is actually a way of detecting oxygen. It's a bit indirect, but you're detecting oxygen by what it bumps into, by the phosphors it bumps into. Now, if oxygen is not there, if there's no oxygen present whatsoever, the phosphor will eventually emit. And this phosphor in particular emits very bright red light that you can see. So what did we do? We decided to take phosphors and actually create a way of looking at oxygenation over large areas in a bandage, and we call this a smart bandage. We actually have a bandage that is a liquid bandage. You can apply it by painting it or spraying it over. And what it does is when you shine blue light on it is it emits. Now, if the tissue is healthy, if there's plenty of oxygen, the oxygen is stealing away all of the energy from the phosphor. So the bandage just emits green light. Green, good. The tissue is okay. If there's not a lot of oxygen, that phosphor can start glowing, and it glows bright, bright red. And you can see that with your eye, and you can see areas of the tissue that actually have low oxygen. And this can be seen by anyone. Anyone can use this, and we've designed it specifically so it's something that's pretty simple to use. So for example, if we were to have a burn, for example, and we were to put, this is actually what the burn looks like if we cut it up and do a biopsy, and we were to put the bandage over top of it and take a picture with a camera, this is actually what it looks like. You can see the full extent of the burn, and by simply taking the red ratio here, by using at that color, you can actually measure quantitatively what the oxygen levels are. And this is something that can be done very easily. This in fact was taken with just a Nikon camera. So the trick, the complexity, the high-techness of this is actually all in the chemistry and making the chemistry work. And in fact, the whole goal at the end of the day is to make sure that we can do the entire thing with one of these. And we just have created new molecules in the lab that actually allow us to do the whole thing with just a simple cell phone. So just to show you it works and stop the red light from blinking, I'll show you. This is actually done in a graft model. This is actually on pigs because we're not yet in patients. We're just about to start in patients this coming year. And if we do two different kinds of grafts, there's what's called a full-thickness skin graft, where we take healthy tissue and we attach it onto a wound. And there's a partial-thickness skin grafts, which are just the top layer of skin, mostly dead, and we attach it to a wound. And we take a look at what the oxygenation looks like. We see that the full-thickness graft, which is what it should be, is very healthy on both sides. Green is go. Now, the partial-thickness graft, which is damaged tissue, is giving us a bright red signal. It's telling us it's not healthy, and the tissue has not taken yet. But if you come back a month later to the same site, it's a little washed out up here, I apologize, but it's totally green. What we see on our camera indicates that the wound has healed well, and that's tissue that is now viable and is useful. Now this is probably a few years out. I'm hoping that this is something that you may see with a clinician in 3 to 4 years, and something maybe that you can pick up in a CVS 5 to 7 years out. So with that, I'll stop because I'm getting a lot of blinks, and I want to acknowledge the people who've provided some of the slides I was able to show, especially Gary, who's taking all that very interesting body camera work in my laboratory. And thank you very much.
Thanks. That was great. So thank you very much, Professor Evans. I would encourage you again, if you have questions, please fill out the cards and pass them to the proctors who are walking about. Now turn to Professor Jeff Karp, associate professor that you heard at the Brigham and Women's Hospital and Harvard Medical School, and he's principal faculty at the Harvard Stem Cell Institute. He's also affiliated with MIT through the Harvard-MIT Division of Health Sciences and Technology. And he really is a pioneer in innovation. His work has had profound effects on basic understanding of stem cells, on emerging new materials that can adhere not only in dry conditions, but in wet conditions. He's started companies, he's taught. He is the real threat across the board, and it'll be a pleasure now to hear his views on emerging innovation. Thank you.
Right. Thank you so much for the nice introduction, and good evening. In my laboratory, I've tried to establish a paradigm, and that paradigm is where we don't just conduct science for the sake of science, for the sake of publishing papers, but rather for the sake of trying to have improvements in the quality of life of patients. How can we develop technologies in the lab that can't just make it to the clinic, but can be rapidly accelerated to the clinic? And I got to tell you that science, especially when you're dealing with biology, is extraordinarily challenging. 9 times out of 10, when we conduct an experiment, it fails. Now, when you try then to take that science and translate it into products that can help patients, the challenges increase by orders of magnitude. So one of the things that I have learned is that solving medical problems is crazy challenging. And I think that one of the reasons why it's so difficult is because of the education system that we've all been subjected to. I think that what happens is when we encounter failure, when we encounter challenges in our lives or in the lab, we actually approach those challenges the same way every time, and we expect different outcomes. How many people have seen the TED Talk by Ken Robinson? I think it's one of the most watched TED Talks of all time, something like 30 million views on YouTube. Everyone, I think, should watch it. It's really amazing. And what he talks about in that lecture is how the education system, how we're all born creative. You know, just watch kids play. But the education system has actually educated us out of being creative. And I think that that actually impacts how we approach problems, how we approach challenges, how we approach failure. And I think it ends up really keeping us on a narrow trajectory. Now let me show you an example of what I think really kind of emphasizes this point. This is a problem. Here we see a car that's fallen into the water. And we have the problem solvers that are lined up here to try to figure out what to do. So they get a crane. And things look like they're going well. But then this happens. So what do you think the problem solvers are going to do next? They get a bigger crane. They come at the problem from exactly the same angle. And then what happens next? So I think the challenge that we and they face is how do you break free from this repetitive thought process? And I would argue that our brains in general like to operate in a very low energy state. It likes repetition. And I bet that if most of you, go home and go into the shower and move where you have your shampoo bottle or your soap to a different location, wake up in the morning, you will probably reach for the location that it was previously and not the new location. So I think there's many ways to actually break free from this repetitive thought process. And one of the ways that we've been trying to do this in my laboratory is to look at nature for inspiration. And it's this idea that every plant, every animal, every living creature that exists today is here because it has solved an incredible number of problems. And those that haven't have quickly become extinct. So in essence, we are surrounded by solutions, ideas for solving problems. And I wanted to share with you some examples of how we applied this principle of bioinspiration to overcome what were seemingly insurmountable challenges that we were faced in the laboratory. Here's one. One late summer evening in 2009, Dr. Pedro Del Nido, who's shown here, who's chief of cardiac surgery at Boston Children's Hospital, sent me this email. And there's an excerpt here. And what he said is that he's treating patients, kids, who have septal defects. These are holes in between the chambers of the heart. And he said, you know, there's devices that have been developed that work fairly well for adults, but they're permanent, they're non-degradable. And a lot of groups are trying to figure out how you can just downsize these. And that's problematic because those hearts are growing. And so these kids are going to need to have revision after revision after revision procedure as their heart grows. And he says, you know, we've tried to suture this, but many times it just tears, and we have nothing available to seal these holes. We can do better than this. He said, can we work together to develop a new type of adhesive that would solve this challenge? And we had been developing adhesives for some time, so he was aware of our work. And so what we did is we set out to develop an adhesive that could work in the most challenging environment in the body, inside a beating heart. And with many of the projects, what we try to do is not just take something we already have and force it on the solution, but we actually step back and say, what's the design criteria for the ideal solution? If we could come up with it, what would some of those criteria be? And I got to tell you that this is really, really challenging to do because it's easy to come up with a list of 20 or 30 things. The challenge is, how do you break it down into 5 or 6 things and then narrow it further to 1 or 2 that's going to completely drive the solution, that you want to be differentiated from what anyone has ever tried before. And so we came up with this short list. We thought, if we're going to put this into the heart, we don't want it to be fouled in the presence of blood. We don't want blood to react with this material and make it so it wouldn't attach to the tissue, which is the current case for a number of glues or sealants that are in the clinic. We also wanted it to be fully degradable, and as this degraded, so we're thinking, we initially thought of a patch. Could we just stick some sort of a patch in there? And if that patch was degradable and cells could migrate on top of it and into it, it could be replaced with the patient's own tissue, and therefore they wouldn't require any revision procedures. It would also need to be elastic because the heart's contracting, you know, it's expanding and contracting over multiple cycles. It would also need to resist washout. There's a lot of stresses, a lot of forces. There's flowing blood in the heart, and it can just wash away. And then we spoke to a number of clinicians, and they said, you know, we'd really like this to be on-demand adhesion. They said, we have a couple glues available to us in the clinic, and you know, they're not that great. They either cure within 1 minute or 10 minutes, and we want to be able to cure it when we're ready. We don't want to be at the mercy of the technology. We want to be in control. So it turned out we ended up having some materials that could address some of these challenges, but we had no idea how to address these two. How could this work inside the heart in the presence of blood and not react with the blood? And how would it remain in place and not just be washed away? And so what we did is we turned to nature for inspiration. And we looked at creatures on the land and in the sea that exist within wet, dynamic environments. And we looked at sandcastle worms in the sea and slugs and snails on the land. And what was really interesting is that these creatures all had something in common, which was viscous secretions that stay put like honey on a plate. And when you look at these viscous secretions, they also contain hydrophobic agents. And hydrophobic agents can repel water. So we thought, well, what if we developed a material that was viscous and hydrophobic, entirely hydrophobic? So when we put it inside the heart and pushed it up against the surface, it would just repel the blood away from the surface. And because it was viscous, it would stay in place long enough for us to cure it. And for curing, we chose to use light activation. There's lots of ways, as you saw from the previous lecture, of how to put light into the body to cure these types of materials. Now, we spent a long time working on this technology, multiple iterations, and I'm going to show you some of the examples to demonstrate that this can actually work. We were able to come up with a material that addressed all of those criteria. And so in the interest of trying to bring this to patients, to improve quality of life, to enable new surgical procedures, we started a company called Gecko Biomedical. We started this company at the end of 2013, and during the past 14 or 15 months, the company has taken the technology we developed in the lab. They've scaled it up to tens of kilograms that they can produce. They've stabilized it, and they're en route to be first in man by the end of the year, perhaps even in August or September. Now, we're not going to go after the septal defects as the first application. We're keeping that in the academic environment, and we're still advancing those studies. We haven't yet shown we can seal the hole inside a beating heart, but we're moving towards that. But this type of material has so many applications in the clinic. We're still using sutures and staples, which have all kinds of limitations, and this has potential to replace them or augment them almost everywhere in the body. Something else I wanted to share with you that I alluded to at the beginning is that I really think that innovation requires being ready to fail repeatedly. Something that's very difficult, I think, to deal with sometimes. And what I've noticed, I've been reading a lot of articles on failure, is that the most successful people end up having the most spectacular failures. Let me give you an example. Steven Spielberg had a movie once, his first movie, which was the Sugarland Express, complete disaster. It had a limited release, one of the worst things that can ever happen to you in Hollywood. And so he had trouble finding work. And he eventually, through a lot of persistence, was able to convince someone to fund a new movie that he wanted to create called Jaws. And he said, you know, I can do this movie, 55 days, no problem, $4.5 million budget. But there was a problem. He relied on a mechanical shark, and it kept failing and failing and failing. And the movie ended up costing $10 million, more than double the initial budget. And it took 159 days to film the movie. Now imagine actors that have dedicated, they've preset the time of 55 days, and now you have to keep them 3 times longer. Imagine how challenging that would be. And to then convince the people who funded this movie to keep adding more and more and more money. This is the kiss of death in Hollywood, if you go over budget this much, both on time and money. But because he was so persistent, because he wouldn't let go of this, something remarkable happened. And what he realized was that actually not showing the shark was more powerful than showing the shark. It was actually scarier to see these types of buoys being pulled in the water, and your imagination's running wild because you're visualizing this shark at the end of this hook that just, you know, has so much muscle power. It's like pulling these buoys in the boat along. And a lot of people attribute the success of the movie to not showing the shark, to really harnessing people's imagination. And it's this idea of the persistence that he had to turn failure into success. Now, I've really tried to implement this in my laboratory, and let me tell you about a failure that I had. When I started my laboratory, I developed this technology. It was a stem cell therapy. And I had really nice, what I considered, data supporting the idea that we had generated. And I took it to Newbar and Dave, who are partners at Flagship Ventures, a venture capital firm, to see if they'd be willing to invest money in this to take it further so we could advance it to the clinic. And they looked at it and they said, you know what, this is really interesting, but this will never, ever, ever make it to the clinic. Your work is too complicated. There's 5 steps in your procedure. You know, it's hard enough to translate something that has a single step. You have like a 5-step synthesis. Like, what are you thinking? There's no way this will ever, it's going to make a great paper. It will never help patients. And I went back to my lab shocked, first of all. But I reflected a lot on this. And I thought, you know what? I'm going to start projects in my laboratory that take this to heart. How can we develop technologies with this idea that we need to be able to manufacture? We need to have batch-to-batch consistency. If we're going to help patients, we can't just think of the innovation in the laboratory. We have to think about what happens later on, the manufacturing. If it can't be manufactured, it can't help patients. And so I started thinking about what problem could we work on, and I looked here at my hand, and you see this kind of red here, and I was like, you know, this is really bothering me. This is a very itchy, inflamed region of my hand. What's going on? It's next to my ring. What's happening here? And I started reading up, and I realized that this potentially is a nickel allergy. And I thought, you know, how could this be a nickel allergy? This is 24 karat gold. That's not possible. So I took it to the lab, and I performed a reaction on my ring. And here you see this red here. This is reacting to show that there's nickel in my ring. And I realized I have a nickel allergy. And so I thought, okay, you know, is this a big problem? Turns out 9% of the population has a nickel allergy. And nickel is something that you're exposed to everywhere. It's almost impossible to avoid. It's in eyeglass frames, keys, coins. It's all over the place. And I looked into examples, and there's actually nothing to prevent this reaction from happening. Once you reach a certain exposure level, you're sensitive to nickel for life. Every time you contact nickel, you're going to have a reaction. And so I started thinking, maybe we could solve this problem. Maybe we could prevent these reactions from happening. Could we develop a prophylactic? And so what I did is I went to a list. There's a list of agents on the FDA website that's called Generally Recognized as Safe, Grafts. These are agents that exist in toothpaste and other consumer goods. And my thought was, these exist in ton quantities. They're extremely cheap. And maybe we could find agents on there that would bind the nickel on the surface of the skin and prevent the nickel from going in. And maybe we could use particles. Sunscreen has nanoparticles in it that block the sun on the skin. What if we could create nanoparticles that you coat on your skin and it binds to the nickel so the nickel can't go through? And so we created a cream that has particles from agents that are safe, according to FDA, that could block the nickel. And here's some of our results. So here we're looking at a full thickness through skin. So if you have, here's the top of the skin and the bottom, if you just take your skin, and we put these particles, calcium carbonate, this is in toothpaste, calcium phosphate, and we put these particles on the top of the skin, and we added, you see the calcium signal here from these calcium-containing particles, and we added lots of nickel. And the nickel bound to the top of the skin, it didn't go in. But if you don't have these particles, the nickel goes right through. And if you wash the skin, the nickel and the particles go away, but it stays if you don't have the particles. You can't remove it. And so we published this paper in 2011, and it was picked up by CNN and LA Times and other outlets, and people started writing in, when can I have this cream? When's it going to be available? And so we started this company called Skintifique. And within about 2 years of starting this company, we were able to transfer the technology into the company, scale the product, and start selling it. And about 80% of the people who have tried this, it works. 1 or 2 application works for an entire day. And so I'm just really excited about these technologies and the potential that things that are developed in my laboratory can make it to the clinic or en route to the clinic. And I think that one of the critical components to this is also having an exceptional entrepreneur, because we're only doing 1 of 10 steps that need to happen in order to help patients, the development of the initial technology. And what I've realized in this process is that there's a long way to go after we publish the paper, after we demonstrate the proof of concept of these technologies. And this is something that I'm committed to do in my laboratory, and I wanted to thank you for your attention and the people in my laboratory and my funding sources.
That was wonderful, two wonderful talks. We'll invite Professors Karp and Evans to join at the table, and I'll read some of the questions. There have been fantastic and wonderful questions. I can't get to all of them, so forgive me. There have been specific questions and there are general questions. I think we'll start with the general questions, and if we have time, we'll go to the specific ones. So the general question for both of you is that many of the people are wondering about the following, and that is, if all three of us are correct that we need innovation to advance medical science, here's two things to ask you. First, how do we promote innovation? How do we incentivize managing costs, especially as this country's expenditure on healthcare is approaching 20% of its GDP? How do we incentivize industry if we insist on costs being managed? And what's the role of basic research? So there are four things for you to consider. Why don't each of you take a minute and a half to answer two of those? And I'll say them again. What's the role for basic research? How do we manage costs and support innovation? How do we incentivize industry to become our partner if we're managing costs? And if you had to choose, I'll recast the fourth one, which of those many things would you prioritize as number one?
I'll just start with a few general comments. I gave a talk in University of Toronto a couple weeks ago, where I did my PhD, and there's a professor there who has developed a technology that is en route to making a major difference in the lives of patients. And he gave a talk in this research day, and he said, you know what? It took me 3 decades of basic research before I was ready to have that technology leave the laboratory, till it was ready to be manufactured and turned into a product. And I think it's important to emphasize that we really need the basic research in place, and we need to have good understanding of the biology and of the materials before the technologists can come in and really formulate and create technologies that can leave the laboratory. So I think the basic research is essential.
So Jeff, I'll interrupt you. That's the easy answer. The more difficult one is, how do you simultaneously fund basic research, innovation, and control medical costs?
I think one of the challenges is that when you look at researchers kind of across the board, there's very few that have actually translated technologies and helped patients. And when you look at the makeup of the review committees for grants, you submit a grant to the NIH, for example, it's not clear to me that the people on the panel actually have expertise in translating technologies. And I think so one possibility would be to try to look out and to search for those people who have been involved in translation and have them review more of these grants to maximize potential to pick grants and fund projects that have potential to make a difference in patients' lives.
So Connor, if industry is essential to innovation, they take a pill and they take a huge laser and they make a medical laser, they take a big rod and they make a pill. How do we incentivize industry if we're trying to manage costs?
Well, part of that probably lies above my pay grade, but I think definitely at the early stages, having industry being involved at the fundamental levels is very, very important. One of the things I think Mass General Hospital and Brigham and Women's Hospital are doing quite well these days is actually to bring industry partners in to our labs to work with us at very early stages, and that stops a whole lot, right? I've had experience very similar to Jeff, where I was developing this bandage, and I had all these great ideas and things I wanted to do, and an entrepreneur group had come in and was talking to me, and they said, whoa, stop. You're going way overboard. That would take you 10 years of research, 15 postdocs worth of time to do, and meanwhile we can take this piece and start rolling with it right now. We can begin bringing this out to patients, and we can bring in the funding to do it. And I think, at least on the research side, having those conversations early and understanding what the outputs can be make the process more efficient. In terms of raising costs or lowering costs on the outset, I think there needs to be, Jeff touched on it, I think there needs to be a very strong push on the government side, on the funding side, to actually incentivize, to de-risk a lot of these early involvements, to bring in companies, to bring in entrepreneurs, to bring in investors that are willing to take a lot more risk to early technologies. It lowers their costs. It increases the amount of things that make it out the door. And in that speeds up that pipeline, so it's not 50 months till we have something approved.
I would also add that I think in general, academics are unqualified to really do things that are innovative, meaning to bring ideas and develop products and actually help patients. We are trained in asking questions and formulating hypotheses and putting together experiments and publishing papers and presenting our work. But really, most of us have no idea what happens when something leaves the lab, what the next steps are. And there's a lot of steps. And if we know more about what happens, I think we can impose criteria early on and pick ideas that have a better chance of making it. And things like patents. If you don't file a patent and have intellectual property and protection of your ideas, then no company is going to want to invest. And there's really an art form to writing patents and how you develop families of patents. Also in terms of thinking of clinical trial design and manufacturing, and how do you pick, often we'll develop a technology and we have no idea what's the best first application. It may be a platform, it could be used in multiple areas in the body, but how do we figure out what's the best first application? And many of the times, it's not obvious, and it requires groups of people coming together who have brought technologies to the clinic and multiple clinicians, and it's a big conversation. But I think if we somehow were able to have academics, especially those who have access to tools and to develop technologies, if we had better understanding of what happens after things leave the lab, I think we could make better decisions of projects to start and pursue that have a chance of helping patients.
So do we run the risk, though, of insisting that all science be innovative, and do we create silos of excellence for the have and the have-nots, pushing down the independent basic scientists at the expense of the sophisticated entrepreneurial scientists?
I think you need both.
You need to have both. And I think the challenge we have right now is as Jeff said, if I take a look at my students, most of the students that we prepare, the people who are going to follow us, are still, focusing on mostly hypothesis-driven research, and that is really important, and it has to stay. It's the bedrock. It's the foundation of what we do as scientists. But there has to be a training. There has to be a pipeline model of bringing science from the very fundamental eurekas all the way out to the patient. And I don't think that necessarily separates have and haves not. I think it's realizing that there are strengths and there are weaknesses to different approaches, and that we need to bring in partners along the stages, at critical junctions along that stage, who can help bring technologies out to patients in an efficient way.
So let's take what you've both said and address some of the specific questions. What in your mind are the two most essential and critical factors that are standing in the way of a cure for cancer, solution for HIV, bringing xenotransplantation to the clinic, any of the big things? What are two factors, two general factors? And my follow-up question as you answer that will be, what would you advise if you were a presidential advisor for us to do immediately?
Wow. I mean, I would turn the question back to
Yeah, I think I might as well.
Dr. Edelman. I think is best positioned to address such a question.
Okay. So, so then I would answer the question in the following way. We need to do everything, and that's impossible. So the most important thing we need to do is to educate. We need to involve all of you. And the fact that you're here today is actually the very first step. We can't do this alone. We need you, as an educated public, to be willing to fund our research. Because at the end of the day, you're not letting us have a good time, though we have the greatest jobs in the world, but you're actually ensuring your future and the legacy of your children and your children's children. So the most important thing that we need to do is project science not as something that goes on in an ivory tower, but rather something that goes on in the clinic, in the laboratory, so that it can rapidly make its way to the community. And the community has to absorb science and innovation with the understanding that it will fail over and over again, and just give us a little breathing room to make it better for you. Second thing we need to do is, from a policy point of view, divorce essential elements of what makes our lives better one from each other. We have a terrible tension that exists today, is that the funding for school lunches and child vaccination and K through 12 education comes from the same budget that funds these investigators. So that you have to make a choice. You have to make a choice between propelling photomedicine or stem cell biology or innovations in the latest molecular imaging and providing lunches for inner city children. We can't do that anymore. It just can't happen. And that needs to involve all of you. We have to set aside funds for everyone in this United States so that we all get the benefit, and it's not that we go to the back of the line once we've gotten the first choice. So those are the first two things I would do. Now I'll turn that back to you.
I'm speechless.
Yes.
Those were perfect answers.
I can't imagine. I've never known you to be speechless. Okay. So I'm going to ask both of you to finish, because we have 5 minutes, in describing how you deal with failure. You both alluded to it. And what is it was your you told us your spectacular failure, but it really wasn't a failure, it was a success. And you told us Steven Spielberg's failure, but what is the recipe for dealing with failure?
So I'd say when I started my faculty position, I started applying for grants. I got my first grant, which was a small grant, but then the next 9 I didn't get. And it turned out that in the first 2.5 years, I submitted 100 grants, most of them failures. But I was determined. And what I had tried to do, there was a problem with my approach at the time, which was I didn't want to do just incremental work based on things I'd done in the past. I wanted to pitch completely new ideas. I wanted to get into new areas. And the reviewers kept coming back and saying, but you have no preliminary data for this. This is too high risk. And so what happened was, is though through the persistence, I was able to turn that failure into a success. And the way I was able to do that was because in many of those failures, those failed grants, I got feedback on the grants from the reviewers saying exactly, putting them together, putting all these failures together, the recipe for how to write a really good grant. And so I'd say that now, when I write grants, I can almost envision the 10 people sitting around the table and know exactly what they're thinking, which I think now maximizes, now we don't get every grant that we apply for, but we get enough that we can still pursue multiple avenues of research. And so I think that one aspect, just like we saw with Steven Spielberg, is having a lot of persistence. The other, I would say, is having access to the right tools, the right technologies, the right people. And one of the ways that we try to do this in my laboratory is having a multidisciplinary team. So we have chemical engineers, mechanical engineers, immunologists, basic biologists. We've had a gastrointestinal surgeon. We've had a cardiac fellow come through the lab, multiple MDs. And so there's very minimal overlap in expertise in the lab. Everybody brings something unique. And when we get together to brainstorm and solve problems, everyone feels validated. Everyone has something to offer. There's minimal competition of the people in the lab. And I think that that has allowed us to come up with lots and lots of ideas from all kinds of different disciplines, and then we can, you know, pick the best ideas to advance. And I think that that's really helped.
I would agree with all those points, and like Jeff, I've probably written over 100 unsuccessful grants myself. I think what a lot of people see, what we show others, especially as scientists, is we're like the iceberg floating in the water, right? All you see is the tip. The success that you see is what sees on the surface. The rest of the iceberg, the part that's beneath the surface, is where all the hard work, the blood, sweat, the tears, the overnights, the late nights, and the failure is. And it's funny because I think one of the things you learn, part of the process of getting a PhD, I think, is a very humbling experience, because you learn that you are going to fail all the time. And I think that's why you find PhDs, why we're the scientists. I tell every student who comes through my laboratory that they need to remember this one thing, that if an experiment works on the first time, something's gone horribly wrong. Because it's never going to work the first time, and it's not going to work the second or the third or the fourth or the fifth, and probably not the 25th, but magically on the 26th, part of it's going to work. And when that happens, that's the moment that you need to sit up and go, aha, why did it only partially fail? Why did only a little bit of it? Where is that insight? And that's where the insight comes from. And I think it's something that we as scientists, we beat ourselves over the head with failure constantly. And I think it's something that people who aren't, and you're going to see this, you go to the best innovators in the world, the CEOs of your Fortune 500 companies, they project success, but they are failing constantly. And it's learning from those failures, and it's basically setting yourself up to fail and fail often. That is probably the most important part of doing any of this work. Now, in some cases, you can't fail. If I'm a doctor treating a patient, you better hope I don't fail, right? But on the research side, this is what gives us this flexibility. And I think it's this mentality, it's this education mentality, that if you're going to try to do something hard, you should expect to fail, and you expect to learn from every single one of those 100,000 failures, because the moment you get it right is the moment you've integrated every bit of knowledge you've learned along the way.
And make failure part of the process.
Yes, it's part of what you do.
So unfortunately, our time is up, but I want to thank first Dean Wild for making this possible, Angela Alberti for organizing this, for all of you for coming, and for allowing us to be a part of your lives and allowing us to do what we love and what is the most wonderful job in the world. So thank you very much for coming.